Plant community composition and diversity. When we measure vegetation attributes on rangelands, we often do this to provide data that are relevant to management. Very commonly, we have management or conservation goals that pertain to a particular targeted plant community, and this can be described as having a certain composition or diversity of species. Diversity reflects the variation within and among living organisms on a site. In this discussion, we will be focusing on plant diversity exclusively. There are a variety of scales at which diversity can be considered, such as genetic, taxonomic, considering the life forms or functional groups, and also landscape scales. The alpha and gamma, beta and gamma diversity are often used to describe differences in diversity at different spatial scales. Alpha diversity refers to within site or within habitat diversity and is concerned with the species and their relative abundance at the site, and so they reflect richness and evenness. Beta diversity is between site or between habitat diversity. So when we're looking at beta diversity, we're comparing usually two sites and looking at the differences in the diversity between two different sites. We can also consider beta diversity to be representative of diversity across a gradient. And if we look at a schematized diagram that shows species abundance as it changes along an environmental gradient, we can examine this concept about the gradient factor in beta diversity. Here we see the different curves represent the appearance and disappearance of plant species along some kind of a gradient from low to high. This gradient could represent elevation, temperature, moisture. It could also represent another type of environmental factor, such as a gradient in herbivory. And what we can see is that some species have a very small range, such as this one here, in which it occurs, and other species have a very wide range at which they occur. If we were to sample at different sites along this gradient, we would find that the mix of species that occur at these sites changes along the gradient, and so we see some turnover. We can see that there are shared species between the first two sites. There's definitely a new species that comes in here that was not found on this site, and that some species have dropped out, but they do share some species in common. And the same is true for this third site as well. There are new species that have come in and others have dropped out. By the time we get to the fourth site, there are no shared species between the first site and the fourth site. And so this is an example of turnover, and we can use studies of beta diversity to examine how species change along a gradient and how much turnover there is across some type of gradient. Gamma diversity is landscape scale diversity. When we look at gamma diversity, we are comparing the diversity of multiple different sites in different locations. If you will, the patchwork of sites or habitats and how their species changes spatially. There are a number of ways of measuring and describing diversity. We actually can't go out and just measure something and measure diversity directly. Diversity measurements and species composition measurements are calculated or composite measurements. We're going to focus on species composition, then diversity indices, and similarity indices. With species composition, we're interested in the relative contribution of individual species as a part of the whole, or the entire vegetation. Usually we use density, cover, or biomass data to calculate species composition. Frequency data are not well suited to species composition estimates. And species composition is always reported either as a proportion or as a percentage. We're going to look at an example using this schematized vegetation that you see in the diagram. In the first case, we're going to look at numbers of individuals or density within this particular site. And we can see that grass is the most abundant in terms of the number of individuals. In order to calculate species composition, what we do is we have to first take all of the contribution of each one of the species and calculate a total number for the site, which is 43. Composition is simply the proportion or the contribution of each individual species to the whole. So, for example, with grass, we would take 32 individuals and then divide that by 43, which is the total individuals for the site, multiplied by 100 to get 75% composition. This is done for each one of the species, and you will see that when we are done, if you add up all of the different individual species composition values, they will total 100%. Here's an example looking at cover. Again, it's the same site with the same species, but cover is a different attribute with different values. The cover of grass was 22%, whereas the cover of the blue flower was only 1%. The total cover on the site was 40%. So composition of grass based on cover is 55%, because that is 22 divided by 40 multiplied by 100. We can see that the attribute that we choose when we are estimating species composition is important, because our results are influenced by the values for the different attributes. For example, the species composition based on density for grass was 75%, but it's lower based on cover, and that's partly because the grass plants are relatively small, even though there's a large number of them. The largest change that occurred was probably because the clover, which was not very abundant in terms of numbers, there's only 2 of them, is a very large plant, and so on a cover basis it accounts for 20% of the species composition based on cover. If we were interested in looking at the relative abundance of different forage species, most likely we would focus on biomass. Now, there are a number of ways of measuring diversity, and we like to use diversity measures that provide us with a single number so that we can assign a number to a site that describes its diversity. The first measure is richness, which is simply the number of species at a site. The next important aspect of diversity is evenness, or the relative abundance of species at a site. We're also going to look at diversity indices, specifically the Shannon-Weiner index, as well as Simpson's index of diversity. But first, let's look at richness and evenness and their relative contributions to this concept of diversity. Here we see 2 sites that are very schematized representations of plants, of a plant community. Each site includes 30 individuals and there's 5 species. They both have the same richness because the number of species is 5. But we can see very quickly just by looking at these that Site 1 appears to be far more diverse than Site 2. And that's because Site 2 is dominated by the purple triangle. There's far more abundant purple triangles than there are the other types of symbols on the site. And so that's the difference, that's the influence of evenness. We would say that Site 1 exhibits greater evenness because the relative abundance of each species is more equitable. Site 2 is dominated by the purple triangle. And so because of that, we can think of not only the relative abundance in terms of dominance, but also that means that the other species could be considered relatively rare. The diversity indices that we're going to be focusing on are the Shannon-Weiner Index and Simpson's Index. And what you see in front of you are the formulas that we use to calculate these two indices. Both of them use information about the relative proportion or contribution of different species to the whole and calculate this in different ways in terms of the relative abundance relative to the whole or the total vegetation. Now I'm not going to go into details about the calculations here because that is covered very well in the learning guide. The thing that's important to remember is that the Shannon-Weiner Index, or H prime, reflects the relative abundance more so than Simpson's Index, which is represented with the letter D. The reason I use a D is because it is a dominance index. It is more influenced by dominant species. We're going to look at an example where we compare two different sites. Now again, we're looking at schematized diagrams of plant communities. In this case, you'll notice that these plant communities are actually quite different. They don't have to have any species in common. The nice thing about diversity indices is that we are able to assign a number, a single number, that reflects the diversity of the site, and it doesn't necessarily matter which species, what the identity of the species are, simply that each species is known and we know the relative abundance of the species. So the fact that there are no large trees on this site does not matter. We can still compare their diversity using these indices. Here we see a table that provides the values for the Shannon-Weiner and the Simpson's Index. We can see that the Shannon-Weiner Index indicates by having a larger number that Site 1 is more diverse. These both do happen to have the same number of species at 5, but for the different species in Site 1, there are more individuals of each species compared to Site 2. We have several species here that are relatively rare or do not have nearly the amount of abundance that the grass does. Site 2 is dominated by grass in terms of number, and so that's why Simpson's Index is larger for Site 2 than it is for Site 1. Remember, Simpson's Index is a dominance index. And we can see very briefly by looking at these formulas that Simpson's Index puts a lot of weight on the most abundant species. Similarity indices are the third type of measurement that we're going to look at that describes diversity. With similarity indices, We are interested in being able to make paired comparisons between pairs of sites. Similarity indices are determined based on examining the number of species that are shared between the sites, as well as similarity of the relative abundance of the species that are found on the sites. Many times when we use similarity indices, we're interested in measuring or comparing the composition of plants on one site to a target or a reference site. Now I have highlighted site A as being a reference, and we can calculate a similarity index between site A and any of the other sites in order to compare how similar they are, both in terms of the species that they have in common as well as their relative abundance. There are a variety of similarity indices available, and we're going to focus on two that are commonly used to describe plant communities: the Czekanowski coefficient and squared Euclidean distance. Again, you see the formulas for calculating these coefficients are in front of you. This is very clearly described in the learning guides, and so we will not be going into detailed explanation of the calculations here. It is worth noting that the Czekanowski coefficient is a similarity index, and it ranges from values of 0, meaning completely dissimilar, to 1, which is total similarity between the two sites that are being compared. Squared Euclidean distance, on the other hand, is a dissimilarity index, and so it ranges from 0, which represents total similarity, to infinity. The larger the number for the squared Euclidean distance, that means that the sites are not similar. In this example, let's consider we're going to be comparing 3 sites, but the first site is a target or reference community. So let's assume that we have 2 sites that we have tried to restore, and site 1 is the target or our goal community, the reference site or the reference state in this situation. Site 1 and 2 share a number of the same species as site 3, but the abundance and the species composition in sites 2 and 3 are very different, obviously, than site 1. So we can use similarity indices in order to make these pairwise comparisons. What I've provided for you here are the number of individuals of the 5 species that we have for each one of the sites, and we can see, for example, that site 3 does not have any of the small blue flower. If we calculate out the Czekanowski coefficient, this data is presented to you in the form of a half matrix. The way to read this is site 1. We're comparing sites 1, 2, and 3 to the sites 1, 2, and 3. So we get a reading of 1, or a measurement of 1 for site 1 to 1, because this is total similarity. What we're really interested in are these numbers down here, where we compare site 1 to 2, site 1 to 3, and site 2 to 3. The squared Euclidean distance coefficients have also been calculated, and they follow the same pattern, where we have total similarity within a site. So site 1 to site 1 is completely similar to itself, gets a 0, and these numbers down here represent the coefficient for site 1 to 2, site 1 to 3, and 2 to 3. As we can see, The largest Czekanowski coefficient is for the comparison between site 2 and 3. This is mirrored by the squared Euclidean distance. This number is smaller, indicating that of these three sites, site 2 and 3 are most similar to each other in terms of their composition. But we were interested in seeing how well these sites have been restored relative to site 1, which is the target community. And so of site 2 and 3, site 2 is more similar to the target community site 1 than site 3 is. And this is reflected both by the Czekanowski coefficient, 0.57, is larger than 0.545, and the squared Euclidean distance, we have a smaller number here compared to the coefficient that was determined comparing site 1 to site 3. So in conclusion, diversity reflects the variation of species or life forms in an area, and it may be considered over a wide range of scales. Species composition, diversity indices, and similarity indices are frequently used to describe the variety of plants on a site, and they are calculated using field data. This allows us to describe and compare the variation in the presence of species, the number of species, and the relative abundance of these species on a variety of sites.